from the University of Illinois, Champaign-Urbana, with collaborators on the SCRI High Tunnel project that we are working on here. And she started her position in Illinois in 2020, so a year after I started, but I still consider us sort of cohort colleagues going through this together. So her position is the same as mine. So she is an extension specialist working with horticultural crop production in Illinois. She is actually in the Department of Crop Sciences, but she's an entomologist, obviously, if you didn't know that. And so today she's going to talk about some of the SCRI work, but just more broadly, her work in high tunnels and biological control. So thank you, Kacie.
All right. You're going behind me. Thank you.
Hold on.
There we go. That's a good one.
First one.
Okay. Fantastic. So actually, today I'm going to talk very little about the SCRI because it seems that I'm doing most of my research in high tunnels right now. But I'm going to talk about some of the projects I've been doing since I got to UIUC. And of course, I am, as she said, especially crops entomologist. And in Illinois, it encompasses a whole bunch of crops. Basically, what I say to people is that if it is not soybeans or field corn, I am responsible for it in the state through my extension work, but then a lot of my research then follows by what growers need. And so I just, pictures of some of the crops that I kind of work with. I also work with peaches, and I do work in open field systems, not just high tunnels. And so what happened to me when I got to Illinois in 2020, actually before I got to Illinois in 2020, I was lucky that some of the extension educators in the state were very excited to have this position filled. So when I arrived, the position of specialty crops entomology extension specialist had been vacant in the state of Illinois for about 3 years. And so the specialty crops commodity group was very upset about the fact that they did not have an entomologist to help them at all. But what that resulted in is when I got hired, they were very excited. And so I hadn't even started. I was still living in Washington State, packing up my house in April of 2020, when an extension educator called me about working with her on some projects, and she wanted to work in high tunnels. We couldn't write a grant just that second because I was going to move across country, but we talked a little bit more, and so we put together a grant as soon as I got to Illinois to work in high tunnels. Now, I had worked primarily in open fields before that. I'm not even sure I really knew what a high tunnel was, at least not really. I didn't have a good sense of what that looked like. And so I got to Illinois, and of course, these are some pictures of the high tunnels I work in now. And Bronwyn Alley is my extension collaborator, and I'll show a picture of her in a little bit. And she works down here at the Dixon Springs Agricultural Research Station, which is about 3.5 hours south of Champaign-Urbana. And it is in the Shawnee National Forest. It's beautiful, but it's kind of far away. And so she wanted to work in high tunnels. And the reason she wanted to work in high tunnels is, of course, she has high tunnels on the property that she does research in. But we had quite a few high tunnel growers in the state, and there was some need in the state to start doing research into different control methods in high tunnels. And Bronwyn had traditionally worked on hort questions. So not entomology related, they did a lot of variety trials and whatnot. And so when she brought me in, she wanted to work on insect control outside of just telling people what types of insecticides that we could spray. And of course, another problem in high tunnels for anybody who works in them is that insecticide recommendations kind of are, if they exist, so many of our insecticide recommendations are for greenhouses or open fields. High tunnels are neither of those, so it can be hard to tell growers exactly what to do about their insect troubles. Now, my background outside of just being an open field researcher is I worked on biocontrol in my PhD. So I already had an interest in biocontrol. I primarily worked with stink bugs in my PhD, and I also worked with stink bugs in one of my postdocs as well. And so I suggested to Bronwyn that we should work on biocontrol in these systems. And so this is not an audience that I have to necessarily use this slide for, but it's always in all my talks where we talk about what biocontrol is and what it is not. And of course, it has to be purposeful. You know, when we talk about biocontrol, I think sometimes there is an assumption that biocontrol can just be, well, there's predators there and they're eating a thing. And so we often talk about how it has to be a purposeful manipulation in order for it to actually be biocontrol. And I include the pictures of the things that that I'm generally using in biocontrol. And again, fantastic audience here who knows what all this is. Now, I will say I primarily work with predators. I do have a master's degree in taxonomy of Burkhanid wasps, but I'm not working in parasitoids generally. So I'm working with predators. And of course, you know, lady beetles are common, Aureus insidiosus, and I work with spiders a whole lot. I also always like to mention here that I understand that biocontrol also includes diseases and nematodes. I don't do that work. I won't be talking about diseases and nematodes, but of course, you know, that's all encompassed here. And so basically for my projects, we wanted to focus on two types of biological control. So today, I'm going to talk about two projects that I've done in high tunnels since I've gotten to Illinois, and they're going to focus on these two types of biological control. Now, ultimately, what I'd like to do, of course, is put these two together. But at the moment, we've sort of separated them out into various projects. And so I've got, you know, some pictures here of some of the bugs that we've ordered, and I'll talk about all that. But first, I'm going, as I'm splitting them up, I'm going to start with augmentative biocontrol. And again, this is going to be a review, I'm sure, but this is where you're adding predators to the environment. And so, you know, there's so many commercially available natural enemies. There's many companies that sell these. And we often talk about this, I talk about this as being more like an insecticide spray, but using bugs instead of insecticides, of course. What we're not doing, generally speaking, is permanently establishing natural enemies in the environment. You're always probably going to have to go in and put more in. And there's two different ways to do this. And ultimately, what we'd love to do is we'd love to do inoculation, where you're really taking a small number of natural enemies, you're putting them in the system early in the season, you're knocking down those pests, and you're not having to go back in and do anything else. What we're actually doing, and what we do in these projects, generally speaking, is where we're, you know, releasing a large number of predators in an attempt to overwhelm the pests. And that's really what we were going to do with our projects. We were doing this inundative biological control. So go back to high tunnels. So with protected culture, which high tunnels are, they're this hybrid of a sort of greenhouse and open field system. We know a lot about greenhouse production and, you know, the differences between these two things. With greenhouses, they are, generally speaking, completely enclosed. Now, of course, we know that things can kind of get in and out of them a little bit, but for the most part, they're completely closed. Whereas high tunnels are these permeable structures that things can get in and out of easily. High tunnels, when it is hot outside, will lift up the sides completely, and so you can just, things can get easily in and out. And so it's a pretty permeable structure. And the other difference between these two types of protected cultures, greenhouses have these really predictable pest issues. If you grow in a greenhouse, you're gonna have thrips, you're gonna have, you know, aphids, you know what you're going to get for the most part. And in high tunnels, you're actually gonna have all those things too, but you also might expect a mix of greenhouse and open field pests. So one would expect that you're going to have stink bugs, you're going to have caterpillars, you're going to have all of those things in high tunnels. So your approach to biocontrol in these two systems are very different. And what we know in greenhouses with biocontrol is that predators are pretty effective, provided that you do it right. Obviously, if you release ladybugs at the wrong time and you try to do those in a greenhouse, they're just going to get out as well. But predators, biocontrol in a greenhouse system is a pretty good control method. They really can't escape, and especially not a lot of the ones we use. However, of course, in high tunnels, they can. So if I'm releasing aureus, for example, it can fly out if it wants to. There's nothing keeping it in there. And so as much as we know for biocontrol in greenhouses, and as much as high tunnels are kind of a like structure, we don't know if this is a viable control strategy for the system. And I mentioned this, and I'll mention it again. We really focused a lot on these pests that were shared between greenhouses and high tunnels. And part of this is because, because of my extension appointment, I take cues from growers in my state. And some of the first things that high tunnel growers came to me about was, oh, I have crazy thrips numbers in my high tunnels. What do I do? I have aphid problems. What do I do? And so what I got is more of these greenhouse pests, as opposed to, oh, I've got stink bugs in my high tunnels, or I have caterpillars, or whatever else it might be. And so when we were going to engage in this biocontrol project in high tunnels, we started really thinking that we were going to focus on these these common greenhouse pests that I knew were common pests in high tunnels as well. So I arrived, wrote a grant to look at using commercial biological control agents in high tunnels. And I was lucky in that we have several facilities that actually have high tunnel, research high tunnels that UIUC has. And so I wrote this grant, and my collaborators, we have tunnels at Dixon Springs Ag Center, which again is in the southern part of the state. And then UIUC has a sustainable student farm. Now it's a production farm, and it's run by Matt Farino there on the right. And so he was my collaborator. And then Bronwyn Ali is on the left there, and she's the extension educator that I worked with in this project. And so we wanted to do this natural enemy release at both of these locations. So they are, again, about 3.5 hours apart, really probably in different climate zones. And so we wanted to look at the natural enemy release sort of across the state, because we thought maybe, given that high tunnels are a permeable structure, that they might really differ amongst those different geographic regions as well, as far as effectiveness or sort of what we were needing to do. And the other picture is actually my technician, Augusta Kilburn, and Augusta does a lot of for this project, I did a lot of the actual work on the ground in the tunnels in Dixon Springs, and she did a lot of it up on campus. So I would drive down and do the actual field work at Dixon Springs, but then she took care of a lot of it on campus. And we do go through and identify all the bugs together as well. So we actually had 2 questions here. So the first one is sort of what you might expect here, is that, so if I buy commercial biocontrol agents, do they suppress pests in high tunnels or not? The second one is actually something that I'm really interested in. So a lot of my research has focused in the past on molecular gut content analysis. So I'm really interested in what things are actually eating. So just because you have predators in a place and pests there doesn't necessarily mean there's going to be a trophic linkage there. Doesn't necessarily mean that those predators, even if you have a pest control, doesn't necessarily mean those predators are the ones doing it. And I've talked to biocontrol companies that have said things like, well, maybe it's the ones you're releasing, or maybe it's that because you're a grower is buying predators and they're expensive, that grower is certainly not going to spray any broad spectrum insecticides. And so you might be conserving more of your resident natural enemies when you're releasing. And so it's hard to say who's actually doing the work here. And so the other part of this is we're releasing these predators, and we wanted to know if it's the commercial agents doing the predation or if it's the resident predators that are already there. So for the methods here, again, we had 6 tunnels that we used for this project. Now, another thing that I need to note about these projects is that because I was kind of coming in over the top of things that were already happening, I did not dictate to either Bronwyn or Matt what kind of crops were grown in these tunnels that I was going to come and put natural enemies in. And part of that is because, again, the Sustainable Student Farm is a production facility. They grow certain types of things. They grow them for dining services. And so their high tunnels are kind of set. What's going to be in them is what's going to be in them. And then when we started this project at Dixon Springs, they had some other variety trials they were doing in some of those tunnels. And so I just let them plant what they were going to plant. And so part of that was because if I was doing this in a grower field, I would have that same sort of situation where I would go there and this is what they're growing, and I would release these generalist natural enemies. And so you can see that our differences here, the Dixon Springs Ag Center, at least 2 of those tunnels were really just polycultures, just many, many different types of plants. And then 1 was actually more just tomatoes and peppers. And then up on campus, they were all tomatoes and peppers, but a lot of varieties. Cherry tomatoes, slicing tomatoes, variety of things. And so we did this. We had 4 predator release tunnels. So we had 2 releases and a control at each location. The other thing to note is one of our tunnels was the 4-H tunnel. So it was smaller and it had a whole bunch of interesting things going on in there. And so just to keep in mind that these are not tunnels that are fully, they're all exactly the same. So, you know, grain of salt and all that. And then we did insect monitoring where we put sticky traps. We collected them every week. And of course, yellow sticky traps are going to attract certain things. We also did predator collections for that question of whether or not the resident predators or our release predators are sort of doing the biocontrol. And so we only collected 10 per tunnel when we did plant counts, which is plenty in the end. And then we also did plant counts where we went and picked certain plants and we counted the number of predators that were present and identified them to the extent that we could. Another thing I want to note here is at least at Dixon Springs, they did spray BT for caterpillar control, because that is something in an organic system, especially in high tunnels, because the BT sprays work so well. A lot of our growers don't end up with the caterpillar issues because they just spray BT. And we weren't releasing anything that would have taken down caterpillars anyway, really. So that's just another note there for this project. So, as I said, this is the other part of this is this pinpointing what the predators are actually eating. And so this is more on the side of conservation biological control, because again, you're really looking at those residents. So you're trying to identify the predators responsible for control. So then you can, you know, bring more of them in. And I do this with molecular gut content analysis. And I actually do it sort of kind of simply, I suppose. So in the lab, we design specific primers. Some of the primers we use are already in the literature, some are not. And we actually do a really sort of quick and dirty DNA extraction. And part of this is, a lot of this is because it allows us to do a lot more predators, because it's much cheaper than some of the other extraction methods. So we use a Keylets resin extraction. You know, all of this is just to show, of course, spiders and a lot of other generalist predators are liquid feeders. No way we could tell what they were eating, even if we tried to dissect them. And so it's a really simple procedure where you put them in this resin with a proteinase K, of course, do the DNA extraction, and then we screen them. Currently, we have done these sets. So we've done aphids, spider mites, western flower thrips, and columbella for a variety of things. And we visualize it on actually a capillary electrophoresis system, which in future, once I get all the protocols done, will allow me to do these all at the same time in a multiplex system. Right now, what I'm going to show you today is singleton PCRs, because that's as far as I've gotten with the protocol. But the protocol to stack a bunch of prey on top of each other will allow me to screen for specific things much faster without having to do next-gen sequencing, because I am not a bioinformaticist. The other reason I like to do this is because aside from the Kaya XL, if you don't have a lot of money to do molecular gut content analysis, but you'd still like to do it, the equipment and the supplies are not all that expensive. So it allows people, other labs, to potentially repeat this without having to do next-gen sequencing and qPCR and that sort of thing. So in the first year, we sort of just scattershot what are common generalist predators. Again, one of the things that kept coming up from growers was thrips are a huge problem. We need to take care of the thrips. I don't know what to do about the thrips. And so we looked at, these common biological control agents that are labeled. Some companies actually label Aureus as specifically for thrips, which I'm very skeptical about. So we did Aureus and we did a predatory mite, and then we actually did release a dahlia, these lady beetles, and they came as larvae. And we released them monthly, and we did plant counts afterwards. And we did plant counts on 1 of every 10 plants. So in a larger high tunnel, that would mean we did more plants than others. And then we released them according to the company's recommended dose. So how the company says to release them. I'm going to caveat something here that I think is really interesting. Biocontrol companies recommend a by feet of row number of predators. They do not care how big the plant is. So I asked specifically, so in the early season when your plant's this big versus when your plant is up to the ceiling, is there a different recommendation? And the answer is no, which seems like there should be. But that's just something else to note about these kinds of things. I think there's a lot about this augmented biocontrol that we don't really have down yet, and the companies will get there eventually, but there's a lot of research that needs to be done, especially with, sort of what the recommended amount, because this is expensive. And if I go to a grower and they've got to do 2 or 3 times this, and it's going to be really expensive for them to do, they're just not going to do it. And so that's something else to keep in mind. But for this project, we just talked to the sales rep. They said, this is how much to do. And so that's what we did. So the results, this is everybody pooled. This is all pests, and this is all of our tunnels pooled. And you can see that, sure, technically, numerically, there's less, but there's not less pests in the predator release versus control. And obviously, I would have liked to see a different result here. We did not. But obviously, pooling all pests together is not really the answer, because sure, maybe there's something else going on in each, individual pest. And there is, and it's better and worse at the same time. So I should note that we knew we were looking at aphids and thrips. This data here, and that other one as well, is our sticky card data. So again, you're not going to get things like stick bugs, because they don't go on sticky cards, at least not really, not yellow sticky cards. So what we ended up with is aphids, whiteflies, thrips, and then tons of potato leafhoppers, which are a problem in tomatoes, and we had tons of them in our tunnels. So we analyzed those as well. And so the first result, that is great. is this one here, aphids. Lot fewer aphids where we release predators. So you could say the predator release actually did have an effect on it, a statistically significant effect on aphids. Same with whiteflies. So our mix of predators, we might assume, did a pretty good job in decreasing those pest numbers. On the other hand, no real effect on potato leafhoppers, although again, numerically, we had more in predator release. The problem right here. So significantly more thrips in predator release tunnels than not. Now, again, feel like a caveat all the time. I'm going to keep caveating. Of course, when I say thrips, I mean all the thrips that are on a sticky card. And I am not a thrips person, and it's really difficult to identify thrips to species. So with all the grains of salt, there could be some predatory thrips. However, what we did is we did take some of our cards and get some sequences for what thrips we were finding at the different tunnels. And there were a lot of pestiferous thrips on there. This is definitely a true thing. And so obviously, you know, my first thought here is, well, Aureus isn't a thrips specialist, but that's, you know, neither here nor there. But so we, you know, when I blow it out like this, of course, good news and bad news. Mixed results here for the actual predator numbers in these tunnels. Now, I should mention this is 2021. I am actually going to show you the results for both years separately because there are some differences there. We did some different things in both years. So the other thing, of course, that was of interest is the actual predators. So this data is our plant count data. So this is where we go through and we note the predators that are actually on the plants. We randomize the plants. We go and, you know, in each row, we go again every 10 plants and we randomize them every week. So we usually go to different plants. And you can see here that there's, again, numerically often we're finding more predators in our release tunnels, but there's no effect of release or not. And if you'll note, I said we released a lady beetle, and you'll notice there's no lady beetle thing down there. That lady beetle, we never recovered a single one of those, not one. And of course, Aureus, when I'm talking about resident predators versus our released, we also have resident Aureus. We didn't mark, so it's not like with that I could say, was that one re-released or was it not? And so unfortunately, you would expect more predators in a release tunnel that you release predators in versus not, and we also didn't see that in 2021. And again, sure, spiders, lacewings, we didn't release any of those. It's not like we weren't doing anything to manipulate those in this experiment, so fair enough. So again, I'll go back to this, and I did look at the Aureus a little bit more. Again, couldn't say whether they were resident or not because we didn't, you know, find any Adelia. We'll talk more about that, and the mites were also a difficult prospect. They were just kind of in there. So I did look this year at the Aureus recovery numbers, very small, and you know, there's no significant effect here, but what I want to note is that at least in one of our non-release tunnels, which is what SF1 is a control tunnel and DS is a control tunnel. In both of those cases, or in SF1, we didn't find any Aureus. We didn't release any there, but there weren't any either. And so we did have some numerically more where we released occasionally, and here for sure. But again, I don't know if that's residents or just, you know, things that we released. We weren't very good at recovering the things we put in there. So it's hard to say if they stuck around or if they died. It's very hot in high tunnels. So the last thing here is I did do some of my gut content analysis, and there's a bit of a story here as well. So we've got the tunnels, and this is how we labeled them. So DF and DY release tunnels at Dixon Springs. DS is the control down there. And these two are the release tunnels on campus, and that's the control over there. And we have a percent of predators that are just positive for whatever pest DNA we were testing them for. And so one thing you'll notice here is this, the Western flower thrips. So I don't have a thrips primer. I have a Western flower thrips primer. And we knew that we had thrips across every location, lots of thrips. And you can see in this tunnel, lots of our predators were eating thrips, just so many. Again, we're still getting more thrips in the release tunnel, but we've got predators that are definitely eating the Western flower thrips. And that's across the Dixon Springs location. None on campus, not a one. Now, I don't think that's because on campus the predators aren't eating thrips. I think that's because for whatever reason, we don't have Western flower thrips in those tunnels, which you would expect to. So again, with the sequencing, we actually kind of confirmed that. So we took a bunch of thrips off of traps from campus and found that we could not find any Western flower thrips on those. Again, doesn't mean that they're not up there, but the result of this for the gut content part of this is that I have designed a primer that encompasses the other pestivorous thrips species that I found in the tunnel. So I'll be using that going forward, in addition to the Western flower thrips one, because that's interesting because it's one of our main pestivorous thrips. And then of course, we always test for columbella. A lot of our generalist predators, and especially spiders, will eat columbella above all else. And so one of the questions always is, if they're not eating your pests, what are they eating? And outside of me doing a lot of intraguild predation work, which I'd love to do, but there's no time, we often use this columbella primer. And you can see that a lot of our predators were in fact positive for columbella. And then the other thing I want to note here is that aphid predation only occurred in release tunnels. Now, again, there were aphids everywhere. I don't really have an explanation for that, but our release tunnels did have significantly fewer aphids in them than the other one. And what we found in our gut contents is that the aphid predation was only present in a release tunnel. And yeah, I don't have an answer for that just yet, but I think it's really interesting given the results that we had with the aphid reduction there. And so this is really preliminary. I won't be showing this for 2022 because I just have this part for 2021. And of course, in future, there will be more pest groups up here. We'll be including the potato leafhopper. We'll be including some other things as well on this to get a clearer picture of the food web inside these tunnels. And going forward with the SCRI, that project, all of this is going to be happening there as well. So I just wanted to kind of show you of the things that were positive. That the other thing that we learned is that a lot of diversity eating these pests in these systems. So here's an example of some of the things, different spiders, dictinid spiders, this little red linophiid, another linophiid, crab spiders, lady beetles, were all positive for two-spotted spider mite. And so we had, pretty good diversity there with that pest. We had a different suite of predators eating aphids. Again, we still found some spiders, small linophiids, again, positive for aphids, almost always are. Some jumping spiders, ants, interestingly enough. Of course, ants tend aphids, but they'll also eat them. And honeydew is not detectable with molecular gut content analysis. So if you do extractions of just honeydew, there's no aphid DNA in there. So if we find aphid DNA in an ant gut, we can be reasonably sure that it's predation and not tending. And then we had some syrphid larvae as well that were positive for aphids. So, again, usual suspects, although interestingly enough, fewer spiders for this particular group. And then thrips, so many spiders, so many different spiders. So we had, a lot of the same spider groups, basically every group of spiders, including trashline orb weavers that you might not expect to be eating thrips because they're not generally walking along plants. And then and then ants again. And so that just gives you an idea of the diversity of the resident predators that are taking down these pests for us. And again, obviously, since I didn't recover any Athey, you didn't see any of that. But we did see aureus being positive for all of those things, whether or not it was a resident aureus or one that we introduced. So the summary of this year's is predators did have a strong effect on aphids and whiteflies, which was great. And that, as I just showed, diverse predators are consuming these pests, including a diverse suite of spiders eating thrips. And numerically more predators on the plants in the predator release tunnel, but again, not significant. And then the lady beetles, we just, we decided not to go forward with the lady beetles again. They are expensive. They come as larvae, and we had to individually place them on plants. It took forever. And again, it's one of those things where when I'm working with growers, I can't go out and be like, I'm going to give this little box and you're going to walk through and you're going to individually carefully place this bug on it. Like, that's just not a viable control strategy. On top of the fact that we never recovered one. Now, that doesn't mean they weren't in there and they weren't doing aphid predation, but it can't be a long-term strategy for me to go and talk to people. So we just had to let that go. And so we replaced them with the other usual suspect. So we decided to go with green lacewings. Because again, we were really focusing on generalists. And then for the predatory mites, we switched it up with sachets. So in the first year, we bought just the stuff in the bottle that you just shake out on the plant. And I'm very skeptical of that too, because if they end up on the ground, they end up on the ground. And so we ended up doing sachets in year 2 for the mites. And then doing everything else basically the same. And so year 2, incidentally, shows us basically the same stuff as year 1. Still have this positive effect of, predator release on aphids, significant reduction, and we still have way more thrips in predator release tunnels than not. And the potato leaf hoppers as well are significantly more in the predator release tunnel too. Now with the potato leaf hoppers, I don't know that we see as much damage to the plant to those. We just end up with pretty high numbers of them. So in future, we may focus on them less. But anyway, this still happening. Actually, it's happening worse in 2022. And then in 2022, I've got a few more predator groups that I'm displaying here. And lacewings, interestingly enough, again, we released lacewings and we had more of them in our control than the predator release. Now, I will say, probably because that lacewing metric is all life stages of lacewings, including eggs. And so we very rarely saw a lacewing larva, which is what we released. And so again, that might be residents again that were already in there. We had significantly more ants in our control as well. But again, ants are tricky because of course ants aren't always a good thing. But those were sort of the 2 effects. Everywhere else we weren't recovering more predators where we were releasing predators. And the same goes, of course, for spiders, we basically had the same amount in each. And even aureus, again, when we released, there was no difference between release or not in our own recovery and finding them again. So one other thing that we have a little bit of information for this year, and I'll show you a lot more information for my next project, is yield, actual yield. So unfortunately, this is 1 release tunnel and 1 control tunnel at 1 location. That's what we have because they actually took tomato yield data. And so in this particular case, we actually did have more marketable weight in our predator release tunnel than not. I'm not sure that tells us anything about our biocontrol success, given that this is not replicated, but it is data that we actually have taken. And so We didn't end up, I don't have yield data for the farm because again, it's a production facility and they do it entirely. They basically don't take yield. They just, you'll have a bunch of pounds that they send someplace. And they have a CSA and they have a farm stand as well. And so this is not, I don't necessarily think that this is a full-on result, but it's interesting that we actually did have a better yield in that tunnel where we were releasing. So again, for 2022, we're, seeing the same thing. Natural enemy release is reducing aphid pest pressure, seems to. Across the board, it's increasing thrips pressure or seemingly making it worse. And then again, we're sort of, this resident natural enemies are really what we're recovering more than our release predators. And that's why I included this picture of the lacewing eggs. We just had so many lacewing eggs often, but very rarely saw lacewing larvae. So did this project and it was, grant funded and we finished it. And so the question was, okay, so augmented biocontrol for aphid control, yes. For thrips control, absolutely not. And so the next question was, well, what's the next step? What could we look at to go at it from a different angle, and especially on the thrips side of things? And so Bronwyn and I worked together to do another grant to look at a conservation biological control question. And so we wanted to look at cut flower strips in high tunnels. And so, of course, conservation biological control, the other one, again, it requires this manipulation of the landscape or management practices to keep and enhance your predators that are already there. And this, people do this a lot of different ways. And of course, one of the ones that we talk about sometimes with growers is just alternating your use and timing of pesticides. So when you have something, especially like a parasitoid that's coming in that's a specialist, maybe don't spray during the window we know it's going to come in and do a thing, right? And then things like polycultures and weed strips, that sort of thing. But generally speaking, it's this idea of these plantings that are going to provide something for predators, whether they be nectar, pollen, and you want to attract and retain your natural enemies before the pests ever get there. You want them in there and you want them to stick around. And so when we talk about sort of flower strips, this is a thing that, again, there's been a fair amount of research on. And one thing that we talked about when we were thinking about the high tunnel is if you do wildflowers, if you do a weed strip, if you do something like this, within a high tunnel, it's pretty high value real estate. It's a relatively expensive growing medium because even though you're not controlling the temperature, some people actually do control the temperature, and sometimes there's a lot that goes into a high tunnel. And so if I was to go to a grower and say, take out a whole row of your high tunnel and put wildflowers there, they might say, no, I'm not going to do that, right? And so we thought if we could, instead of doing wildflowers, do cut flowers that if they could also sell that alongside their other produce. And so we wanted to come up with a commercially viable product for their high tunnels. And then of course, the con of this is this may not work for natural enemies. And also it might actually increase pest pressure. It could bring in more thrips because there's no reason to think that a thing that has nectar for a natural enemy, why wouldn't a pest also want that? It's not just, we're just bringing in spiders and nothing else. And so it could actually entirely backfire on us. And especially considering we have this crazy thrips problem to begin with, we could just make it way worse if we did it this way. But it was something we wanted to try out and see if we could, use these to bring in more of those resident natural enemies. And especially since we saw a lot of the predation that was happening was in fact the resident natural enemies, as opposed to, of course, the things that we were releasing. And so again, we kind of had these two questions. Does adding a cut flower row increase immigration and retention of resident predators? And then the other one that I have data on today is, does a cut flower row in a high tunnel affect tomato yield at all? Is there a treatment effect on actual tomato yield? And so again, kind of some of the same people again. The difference here is that we have a new tunnel at another extension station in the southern part of the state. So a new collaborator, Maggie Ray, helping us down there, as well as the tunnels. We couldn't really do the cut flower strip tunnel in the 4-H tunnel. 4-H tunnel is kind of spoken for, and so we didn't have enough tunnels to really do manipulations at Dixon Springs, so we did it at this other extension station to have another one with cut flower strip. And then we did it at the Sustainable Student Farm again, and this time I made him plant cut flowers in 2 of the tunnels. So we had 2 treatment and 1 control, both in the south central Illinois, just like the augmentative biocontrol project. And so we did a few things. We actually did in the south, we put the cut flower strip in the middle of the tunnel. And then in the central Illinois tunnels, we have it on the outside of the tunnel. And I haven't really messed around too much. This was this year, so I haven't really looked at how that's affecting things necessarily, but that's how we've divided them up. So they're really 2 different experiments in a lot of ways. And we have 4 tunnels that have a cut flower strip in them, and then of course 2 control tunnels. And it's basically all the same type of monitoring, except that in this case, we also added tomato yield. So we did yield counts of tomatoes. I took a section of each row and I would go in and I would do that. I did that twice a week for about 2 months. I looked at a variety of things. So when it comes to the yield, the way that I looked at it was we have full on marketable yield. There's no blemishes. It's beautiful. It's perfect. Fantastic. Then I looked at this different type of damage. And again, with the caveats, some of the damage that I was counting, especially sometimes the thrips damage, actually does not make the tomato not marketable. It was important for us to find actual insect damage on the fruit. And so it's not a marketable versus unmarketable necessarily, but I counted every fruit. And so trying to then assess whether or not each of those was technically marketable or not, I just didn't. So I had marketable fruit, I had anything with plant bug damage, and then thrips damage, and then caterpillar damage, which, as you can see, sometimes was really, really obvious as the caterpillar was just hanging out in that fruit. The plant bug damage is something that I need to go back and look through a little bit more. Obviously, you can see this is clearly stink bug. I mean, it's stink bug damage, it's stink bug eggs. This is stilt bug damage primarily. And so what we ended up with is lots and lots of stilt bugs, so many stilt bugs in those tunnels. And I mean, I could watch them do this damage. So the stink bug damage, it seems, and I didn't really separate them out because I couldn't be 100% sure, but the look of stilt bug damage is much smaller, more pinpointy, and stink bug damage, is more spongy, if you will. And since we had cherry tomatoes up on campus, we had many varieties of them, and some of them, again, like this, is going to be just covered in spots when it gets red. And you can kind of see some of those out there. So this is the type of damage I was looking at for the yield in these tunnels. The other thing, and I'm not actually we haven't done any analysis of it, but what we did with the cut flowers is we assessed how many stems were marketable, and then we made bouquets and sold them at our student. The students have like a farm stand on campus, and we sold the bouquets. And so we're looking at that as well, with how many bouquets we were able to sell and whatnot, and how many hours my technician spent, about 2.5 hours a week making bouquets. So that's something else to take out of it. When they were just stems, no one would buy them. So from a grower perspective, of course, you're not just going to cut these and just take them to a farmer's market. You are actually going to have to do an extra step there to make this an economically viable thing. So this is my yield damage, or yield data. And I did actually separate this out. So I have 6 tunnels. 2 of them the yield data isn't really done yet. And so I have in color, your cut flower rows are in blue, control rows are in orange. DF and DS are the southern tunnels, SF1 and 2 are the ones on campus. And so we did have a significant impact of plant bugs. There was significantly more plant bug damage in the cut flower, even if I put everything together, there is, but it's primarily driven by this one tunnel that had an insane amount of stilt bug damage on those tomatoes. Now, because the tunnels are different sizes, I have different cultivars of tomatoes, this is all proportion of tomatoes harvested, because just raw numbers wouldn't have made any sense, because especially now with cherry tomatoes, obviously you're getting a lot more cherry tomatoes out. So this is actually proportion of the tomatoes harvested to try to normalize that, to make that a comparison that was fair. And the stilt bugs, again, just so many. And so we actually increased, at least in this tunnel, which again, we didn't down here, but overall, it looks like flower strips make plant bugs worse, but it's really just in this one tunnel. And then that tunnel, as a result, had fewer marketable tomatoes as well. And there was more thrips damage in our control on campus than there was compared to, you know, the other tunnels. And then there was no effect of anything on caterpillar damage. And one thing that I think makes this difficult, and that I will be able to change next year, because I'm actually able to dictate to Matt what we put in those tunnels, is that these tunnels, at least on campus, the ones here are the same cultivars. The difference here is one has flowers and one doesn't. That's the only difference between those tunnels. Whereas the difference between SF2 and SF1, both of those tunnels have slicer tomatoes and cherry tomatoes, but they're entirely different cultivars. There's different numbers of all of them. It's hard to compare the two. And the third tunnel, the reason I left it out is because it's entirely different from all the others right now, and I'm having trouble, you know, comparing. And so next year, when we do the second year of this project, we're going to be able to force these to also look identical to each other. And so I think that will help to really tease apart whether or not we're really getting an effect of flower strips, or if it's just something about this tunnel, it was just full of stilt bugs. And I do have data on pest number and some of that stuff that I have not gone through yet for this project. So all I have for this project right now is my yield data and my natural enemies. Now, I will say, I was, of all the answers, the cut flower strip tunnels had significantly more spiders than the ones without it. And I kind of knew that because when I was in them, they were full of spiders. And when I say spiders, I mean spiders literally on the plants, oftentimes sitting in the middle of where the aphids or thrips actually were. And just so many spiders. I've never seen so many spiders on tomato plants as I saw this year. It was delightful. Spiders and spider egg sacs and just so many. And so we actually did see that effect here, where when we had cut flowers, we had more spiders. And so I'm hoping that next year, as all of these tunnels are more, you know, similar to each other, that we can really tease apart what's happening here. And again, I collected a lot of spiders. So the next step there is to actually see what these spiders are eating as well. We didn't have an effect of any of the other predators, but in every case, numerically, we seem to have more of those predators where we have cut flowers versus not. And so while I can't say, oh yeah, cut flowers are increasing yield, which would be, of course, fantastic. And right now, because I haven't gone through that data, I can't say what they're doing to the pest issues in this particular experiment. I can say that it seems like they're good for spiders, which is fantastic. And so, you know, a summary of this part of it is just that we did have, you know, numerically more predators and especially more spiders. And it doesn't appear that yield is enhanced by cut flower strips. I maybe wouldn't have expected it to be. And the other thing that I'm going to look at is I have the yield data is by row, by section. And so looking at if there's like more damage nearer to the flower rows or further away, and then how that compares both where we have the flower rows in the middle and where we have them on the outside as well. So there's a lot of things I still need to look at here. I did complete my, we did complete the field work for this about 2 weeks ago. So it's been a fun summer. And again, this plant bug damage we saw was driven by one tunnel. And so we need to do our cut flower assessments. We need to look at yield. We need to look at how many we sold and what that means. And then, you know, future direction would be to combine, obviously, the augmentative and conservation biological control together if we find these flower strips make sense to go forward with. And so I suppose big conclusions again seem to be our augmented biological control does reduce aphid pressure, but it makes thrips pressure worse. So, you know, double-edged sword. And our cut flowers do increase our spider numbers on plants, and that we do have a pretty diverse suite of predators that consume our pests, thrips especially. And, you know, what I sort of see this going forward, obviously there's more questions to be answered, but that some of these techniques obviously are just part of a program. I am skeptical that biocontrol itself in high tunnels is the full answer, but as part of other control strategies, especially for aphids, I think it's probably something that going forward, hopefully I'll be able to tell growers about. At the moment, it isn't, you know, what we've been doing, and especially for thrips, it wouldn't be something I'd be recommending just yet. But, you know, in future, as we get more information going forward. And so with that, I didn't really talk about my grad students much because none of them are on these projects. These are the projects that I've been doing, or my technician, because for a while it was just me and Augusta. And our funding is through Illinois Extension. The first project was an extension collaboration grant that they specifically give for when you're working with extension educators. And then the cut flower is funded through a grant from the Illinois Department of Agriculture. And then again, SCRI, it's all that's all part of it, but none of this was technically SCRI just yet. And with that, I will take any questions. Yes.
So when you're doing when you did the cut flowers as a refuge, you mentioned that, you know, this is expensive real estate in the high tunnels. So I'm curious, why not just plant them adjacent to the high tunnels, considering that they're, you know, you said you had regularly had them open because of environmental concerns.
Yeah, so that was a different question, I think, to be fair. We thought about that. We wanted to see what it would be if they were actually in the tunnels, because yes, you could just do things on the outside as well. And I think one of the challenges is that often that buffer of natural enemies coming in is not very long. You know, when people do these experiments where we've got like a big field, it's like, well, you see an effect a little bit into the field, but then it doesn't go very far. And so our thought was, if we actually put it inside, maybe that brings them into the tunnel, and then they, even on the outside, it might get them through the tunnel, potentially. And so that was kind of the idea. And the middle, you know, is also, obviously they're not coming directly from the outside, but getting them then in the middle to stay in the middle is kind of why we did that. But no, that's a really good question. And I think that's, you know, as far as wildflowers, if you were doing that outside of a tunnel, I think that's a great thing to do as well. Something that's really low maintenance. We end up at our tunnels with lots of large weeds outside the tunnels sometimes. And so wildflowers would probably be a great answer there as well. Yes.
I remember you said thrips are pretty much expected predators in a lot of greenhouse environments. Do you think that these high tunnels are combining the pressures of a greenhouse population of thrips and a normal field population of thrips into one location?
I mean, they could be. Yeah. I don't have anything to compare it to because I don't really do greenhouse work, so I don't know what numbers I might expect, but I have no reason to think that they wouldn't. And they're just always in protected culture. And it's probably different species. which again is, I probably need to learn thrips, but they're really difficult to identify. But yeah, they absolutely could be just worse because you're getting them from all sides, for sure. Yes.
Maybe you said this, but the thrips increase in the first part, what's your explanation for that?
Well, I haven't really come up with one yet. I've been thought about it a fair amount, and there's other research that kind of shows this a little bit. I can't remember where I read it, and I wondered if maybe there was the natural enemies are going after other things instead. We put so many predators in there, and so the thrips are somehow released from that. I don't know really, and it's, I don't know. It's that one. I wanted to come up with an answer to that, and I haven't really been able to as much just yet. But yeah, it's really consistent, which is a problem. And I wondered if too, maybe we release these things, and sometimes I think maybe we've got an interguild predation problem. So of course I release all of them at the same time. I tried to release them in different, like not on top of each other, but interguild predation is so common, and so maybe because the predators could be going after each other. Now, why that would only affect the thrips and not the other things, I wouldn't have a good explanation for that, but that's always a concern I have, especially when putting a bunch of predators in a place all at once, is that maybe part of the reason why we're not recovering them as much is that's actually what's going on as well. Yes.
Did you identify the thrips species on campus when you did the?
So there were several of them. Yeah, so there were other Frankliniella species, and there was Neo something. I can't remember. What?
You got soybeans nearby?
So we don't have soybeans as close. I mean, nearby-ish, but there aren't any actually on either of the farms. So they're a little bit further away, I think. Yeah. So that's, I did, and I can't remember what they are now. But yeah, we pulled a bunch off and got sequences. I mean, I didn't identify them myself, but we got sequences, and I feel reasonably confident about the GenBank pool of thrips DNA, because sometimes that can be a little tricky.
Did you ever find tomato spotted wilt virus associated with any of your tunnels?
So maybe this year once, might have had one plant that had it, I think. But no, not really. We don't have a lot of disease issue in the tunnels, weirdly, not yet. Part of it is at Dixon Springs up until we started doing this, because we're trying to look at the spiders in the winter as well. At Dixon Springs, they would steam the beds every year. And so there's like no disease pressure, but there's also a lot of not other things. And so now that we're doing this, they're not doing that down there. And the tunnels on campus are movable. So we switch them every year. And so that might help with some of the disease pressure that should build up in high tunnels, but we don't seem to see it a lot at both our locations. So it'd be interesting to see if it starts building up at Dixon Springs now that I'm not letting them steam the beds.
There's an online question. Do you think the difference in watering can impact the difference between indoor and outdoor pest pressures? Often outdoor environments experience rain or overhead watering, but high tunnels usually have drip lines. Could this have an impact on the types of pests and the feeding habits of predators at all?
I mean, I would think so. I mean, the whole rain happening or not happening too would almost necessarily change some of what's going on, and disease pressure and everything else. I would imagine would definitely affect things. I also think, because I haven't looked at the types of spiders you find on the leaves in open field tomatoes versus the high tunnel tomatoes, we get a lot of spiders that are like on top of the leaves in like a little webbing in high tunnels. And I'm not sure if those are also common on open fields, or if it's more protected inside the tunnel, so there's more ability to do that than there is, in an open field tomato.
I was wondering, if we're thinking about the thrips and the leafhoppers and something that's different from the other pests, I think that's relevant for natural enemies is for my own struggles, you know, their eggs are in the plant and then they have life stages in the soil.
Mm-hmm,
So, but you said they steam the beds.
mm-hmm.
I guess, I mean, might you just use one thrip against another thrips? I think that, don't they market like predatory thrips for mite management or like, anyway, not that you want more thrips, but.
Right, yeah, I don't know. I don't think you can buy predatory
Oh.
thrips yet.
Okay.
Yeah.
Well, there goes that idea.
Yeah,
So much
there's,
for the silver bullet.
well, yeah, and it's interesting the things you can buy and the things you can't, which I think is just what can you rear on a large scale and what can't you? Because we sell sort of the Aureus lacewing thing, which I don't know. Often I'm not sure how effective those things actually are, but they're the main thing that everybody buys. And you know, I think this experiment and some other things definitely keep showing me that it's like, well, this is really expensive and I love the idea of it, but as far as an actual control thing, I don't know. For the ones that we're actually doing, right? Like parasitoids, cool. But I don't know. I really like General's predators. I've been working with them a long time, and it just seems like because we know they won't just eat the thing we want them to eat. They'll eat anything. I mean, it's like spiders. I love spiders, but man, they'll eat other spiders or anything they encounter. And so I think some of this gets really tricky, but I have a lot of growers that are interested in biocontrol. And so got to try to figure out if it's a logical thing to actually recommend to them. Yes.
So when you were doing the molecular content analysis, I'm curious, like, if you know if there's a recovery efficiency between liquid versus solid feeding insects, the like quality and amount of DNA you're recovering?
I don't know. I don't know. I know what we've done in the past. So most of the things that we end up doing with these General's predators are often, you know, liquid feeders. Again, spiders are, Aureus's, you know, that sort of thing. I guess amount of DNA or what we focus on in sort of the gut content world, or at least used to, is the decay rate. So how long would that DNA be viable in the gut of a predator? And of course, that's all over the place. And that's another, it depends. Especially with spiders, like sometimes you can detect it for 72 hours as the half-life. And sometimes you can detect it for 8. It just depends. It depends on the primer. It depends on the predator. It depends on the pest. It's a variety of things. So as far as, that's not answering your question because the answer is I don't know. But that's the thing that we often focus on, is how long is that DNA detectable per that primer in the gut of a predator.
I'm pushing your time. The stilt bugs.
Mm-hmm.
So they're omnivores,
Mm-hmm.
right? I
Mm-hmm.
always think about them in the context of eating caterpillar eggs.
Mm-hmm.
You talk about them as being a pest on
Oh,
the
man.
fruit. So do you include them in your gut content analysis? Like, I guess, are they causing way more damage than eggs that they're eating?
Yes, I believe the answer to that is yes. So we did, in the beginning, we started collecting them. And then in one year, at some point, I started noticing that, like, I could see them on the tomatoes causing that damage. And then this year, it was really bad. Like, I could just sit there and watch them just feeding on the tomatoes. And so whatever else they're doing, and in the tunnel that they were really bad, the one that I showed, we have a variety of different cherry tomato varieties in different colors. So we have green ones and pink ones and red and purple. And boy, those colors, the orange, you don't really see the damage. Like if you just plant orange tomatoes, it doesn't matter how many stilt bugs you have, it doesn't change the texture of the fruit, so who cares? But if you're planting purple or green or yellow or red, boy, oh boy, they're just spotty. And the purple look ridiculous. And so whatever they might be doing otherwise, they're way worse for the tomato yield. And actually that's true on like the big tomatoes. The big pink ones were covered in that same type of damage. But I couldn't really separate, other than literally watching them do it. But like, I couldn't say for sure, especially on the big tomatoes, that stink bug, stilt bug.
But are you gonna look at their gut content? Do you have some in the?
So I have some, we collected some for the earlier years. I'm doing more of like counting them, and seeing, you know, how many were there, how many we collected, that kind of thing. So I wasn't probably, because with the only 10 per tunnel, we kind of get that exhausted pretty quickly with like
Yeah.
spiders and other stuff. Sometimes not. I will tell you, some of our tunnels in the SCRI part, where we have people who are spraying a lot, sometimes you get like 3 spiders. One time we found one of our tunnels had nothing alive in it at all. The guy is a scorched earth grower, but that's another story. But yeah, most of the time in our tunnels, it doesn't take much to find 10 predators.
Well, I know we collected stilt bugs for you this year to do gut
Okay.
content, because we saw a lot
We got
in
stilt bugs for
September
you.
in particular.
Okay.
But when we put flowers in the high tunnels a long time ago, when I was postdoc,
Mhm.
stilt bug was the one we saw. One of the groups where we only found them in our tunnels that had flowers. And I didn't pay attention to damage on fruit, and I immediately thought, oh, they're good, they're preying on caterpillar eggs and summer.
Yeah, so it'll be interesting. I'm gonna really, really, really focus on them next year as well. But yeah, that damage, and the thing about that damage is on a farmer's market situation, a lot of people are much more forgiving of certain types of plant damage, and the stilt bug damage really, some stink bug damage does change the texture of that fruit, right? And it looks so crazy that you're probably calling that unmarketable. But a lot of the stilt bug damage, you wouldn't necessarily have to, and provided people would buy it. I think the question is often people will come up to me and say, well, I would eat that. And I'm like, yes, but would you buy it? So like if a grower has a thing of cherry tomatoes and they're all covered in dots, it'll be fine. You won't purchase that one, you'll purchase the one next to it that doesn't. And so even though I would like people to not care about that, they probably do. So that's tricky.
Thank you, Dr. Athey.